I would like to start by thanking Ronchi for inviting me. I'm very excited to have the opportunity to show some of our data here. My name is Paolo Ronchi, and I work in the electron microscopy facility of the EMBL in Heidelberg, Germany. And here, I've been working here for about 10 years. And in more recent years, my main occupation has been volume electron microscopy. And I'm going to show and share with you some data today about volume correlative light and electron microscopy and how we use these to target fluorescent cells in heterogeneous biological samples. So, as I said, I'm currently most involved in a lot of volume EM methods, but the one I'm going to show today is the focused ion beam scanning electron microscope, so the FIBSEM. And you can see this is the model we have at the EMBL. It's a Zeiss crossbeam 550, and this is the microscope that's produced the data that you will see later. I'm not gonna talk too much about the microscope itself. I guess most of you are familiar with it. The idea of the focus of the FIBSEM is that you combine imaging with a scanning electron microscope and ablating of thin slices of material with a focused ion beam. And in this way, by reiterating the slicing and imaging, then you can obtain a stack of images. So I'm going to show this in this animation. So we open a trench, we take an image, and then we consequently cut a thin layer of material, take another image, cut another thin layer of material, take another image, and so on and so forth. So in this way, we generate a stack of images that can be then reconstructed to look at the 3D landscape.
Excuse me, Paolo?
Yes?
I'm seeing your face, not the screen.
Oh.
Is that the same for everyone?
Is it?
I can see the slides.
I can see the slides.
Okay, carry on. Must be something I'm doing.
Okay, sorry. Okay, and this is then the kind of images we would get from FIBSEM experiment. This is a Nematoxella vectensis, so an anemone. And here we can see a little piece of it. So you see the volume of the FIBSEM is normally restricted to a relatively small volume, but we can get pretty high resolution in X, Y, and Z dimensions. So now, especially with modern FIBSEMs, the acquisition are fairly automated, but there's an important point. So there are important considerations. So the first one is that typically the samples that we prepare are very big for EM scale, and this is even more true for the FIBSEM because FIBSEMs are very limited in the volume they can acquire for a number of reasons I'm not going to discuss too much in detail here. But I'm happy if you want to discuss this later to have some more discussion about this. And the other important thing being in a facility is that these acquisitions are typically very long and also expensive for our users. So we have to try to limit the acquisition of the volume that we want. And then the best way to do this is actually to target the acquisition to the region of interest, so to a region that for us or for the collaborator is interesting. So instead of acquiring very large volumes and very large data set, we try to acquire as small as volumes as small as possible, but in the region of interest. So how do we do this? And this can be actually pretty extreme. So I put down some scales here, for example. So now typically our sample can be 2 to 5 millimeter wide if we use high pressure freezing, for example, and then maybe a few hundred microns thick, couple of hundred micron thick, and then the maximum we can actually acquire with the FIBSEM is on the range of few tens of microns, so not more. So then this explains why we need to target the acquisition. And then how do we target the acquisition? So in our facility we have different approaches depending on how and what are the features that drive your targeting. So when we have anatomical features that we base the targeting on, we typically use X-ray. So we have a small X-ray machine, benchtop X-ray machine in the facility with which we can actually visualize the heavy metal stain sample inside a block. And in this way, we could target specific subregions inside the block. This is an example that you see here with some sea urchin larvae, where we were interested in acquiring a small portion of them. And then the second thing is when we have something fluorescent. So now we cannot rely on the difference of the structure because of anatomical features, but we have a fluorescent marker that that defines where we want to acquire. And this is actually a more tricky situation. And the reason why it is more tricky is that typically for volume EM, as you may be aware, we use a lot of heavy metals to increase the contrast. I mean, this is already true for most of the EM techniques, but it's even more true when we want to go for volume EM, and especially for FIBSEM, because we don't have the chance of doing post-staining of the section. So we would have to include all the necessary contrast during the sample preparation in the block. But typically, when we introduce heavy metals in the sample, we are quenching the fluorescence. And then because of this, most volume correlative light and electron microscopy protocols see the fluorescence microscopy comes before the EM sample preparation in the so-called pre-embedding CLEM. But when you do this, I guess as EM specialists, you're all aware that during sample preparation, we are introducing quite some dramatic distortion in our sample. And even worse, this distortion is not really shrinkage, and distortions are not really isotropic. So we may have parts of the samples that behave differently in shrinkage than others. And therefore, the precision of the correlation between the two volumes, so the light microscopy pre-embedding and the final EM imaging, the correlation precisions can be very low. However, we have worked to try to improve on this aspect, and actually the approach we have chosen is to try to preserve the fluorescence during the sample prep for EM. So we have now a post-embedding volume CLEM approach, and this goes more or less, I'm going to briefly detail here. We have a sample preparation that relies on high pressure freezing and freeze substitution. So we have a specific freeze substitution protocols that I will show later, and is based essentially on using only uranyl acetate as a contrasting and stabilization agent, and then embedding in low weak relation 20. So then once we have prepared the block in this way, we would bring the block to a confocal microscope where we can then take a map, a 3D map, fluorescence map of the block. And then here we will identify a specific target based on its fluorescence and shapes and whatnot. And then once you have identified this target, then we are able to define its position and its coordinates within the block. And then what we will do is, first of all, we are going to trim from the block surface the measured depth of the structure of interest. And then once we have done this, we used 2-photon branding to actually draw on the surface of the block a little frame or some landmarks that then can be easily visualized at the SEM, and then this would drive then our FIBSEM acquisition. So I'm going to show you this more in detail, and I'm going to show you a project that was essentially done in collaboration, actually mostly done by Karel Moker. So she's a student in Yannick Schwab's team at the EMBL. And what she wanted was to reconstruct, to have a full volume EM acquisition of an entire dinoflagellate cell. So this is a plankton cell that they were sampling in the sea. And so now the problem is that as she had environmental samples, the samples were very heterogeneous. So you can see an example here. There are cells of very different sizes and shapes, and she was interested only in a subfraction of them, actually in a specific one of them. But the good news here is that all plankton cells, when you look at them, they have characteristic shapes and also especially a characteristic autofluorescent profile. And we decided to use the method that I briefly showed you before to actually identify and target the cell of interest, Carrel cell of interest within the collected sample. So then again, I put this here so that it's as we have the recording of the presentation, it will be easier for you to actually look at the free substitution protocol later on. So I won't go too much in detail explaining it, but the sample preparation was done by sampling and collecting samples at the sea. So there were nets towed from a boat in the Mediterranean Sea in the south of France. Then there was some filtration through which we could collect a specific size fraction, and we could also concentrate the plankton. And then we were fortunate enough that Leica borrowed us a high pressure freezing machine, so the Leica EMIs, and we could bring it actually at the marine station, so very close to the sampling site, and we were then able to freeze the sample very, very early after collection. So all of the experiments we've done were frozen between 1 and 2 hours from the collection at the sea. So then after high pressure freezing, then we went to free substitution. And as I said, here is the protocol. We only use 0.1% UA in acetone as a cocktail. And then, yeah, so we have a very mild protocol in which we introduce the contrast in the sample, and then eventually we embed into low weekly HN20. So then at the end of this, we had to do some quality checks. So first of all, we had to see whether the fluorescence was preserved after this kind of sample prep. And here I have taken some snapshots of our acquisition of the fluorescence in the block. And you see different fluorescent channels and different cells fluorescence in different channels. And also from a bright field, we could also get very nicely the full shape of the cell. So then we could confirm that the prep was good enough to preserve autofluorescence, plankton autofluorescence in the block. And then the other thing we checked was whether the EM quality was also good enough for what we wanted. And so to this aim, we cut a few sections and we screened them at the TEM, which was much actually faster than screening at the FIBSEM. And then we could see that the cells had enough contrast, so we could visualize organelles. So here, for example, a nucleus for these cells, and here some chloroplasts and so on. And if you zoom in, then you would also have a nicer view of all the other organelles in the cell. So now after this quality check, then we were ready to actually perform the experiment as I outlined it before. So we first brought the block. So here you see an image of the block. You see there are lots of small cells, very heterogeneous in size and shape inside the block. And then this is our starting point. So we have these very heterogeneous cells, and Carrel had actually a very few cells inside this big amount and big soup of different cells that were interesting for her. So now how do we find them? And the problem is that we have a fluorescent cell that now we know that the fluorescence was preserved, so we can identify this fluorescence inside the block, but then we need to determine actually all of the coordinates of this specific cell inside the block. And what we do is actually to place our block in a glass bottom dish with a little drop of water. And then we do a confocal scan. So this is actually a tiled Z-stack. Then I'm going to play here at very low resolution where you see the characteristic fluorescence and shape of the different cells. Now, if we overlay also with the bright field, then we could actually have, when you zoom in and have the really high resolution images, then we could identify the cells of interest. And then now I'm going to show you here how once we identify the cell of interest, then we zoom in the area where the cell of interest is, and then we take a higher resolution map of this area, and now you see the quality that we can get. an important thing that will be that I will explain later. So here we acquire a few fluorescent fluorescent channels. In this case, a red fluorescent channel where we could get the chlorophyll autofluorescence, and a green channel where we could get other kind of structures inside other cells. And the other thing that you have seen at the beginning of the video, if I play it again, is that there are these cyans flashes, and this is actually the reflection that we image. So we don't only image the fluorescence, but we also image reflection, which gives us an indication of where the surface of the block lies. And this will be now perhaps more clear. Oh, and then from the stack, then we identify the cell of interest, which is this one with the red square around it. And now, if I show this in the different views, so we have now our cell of interest in XY, but then we are also able to look at it in different projections. So we have an XZ and YZ here, where we can see the cell of interest, but also we can actually visualize the reflection of the cover slip first and of the surface of the block also. And what this allows us to do is then to measure the distance of the cell of interest from the surface of the block. So exactly, so this is what we do here. So we have our XY view, and then we can see and measure the distance of the cell of interest from the surface of the block. And then with this information, then we would go to the microtome and remove the predicted distance to actually bring our cell close to the surface. This can be done in several iterations, because the further your target is from the surface, the less precision you will have in this measurement. So you can have an example here already at 20 microns, which is not that far. Actually, our prediction was already, it had quite an error. So for example, here, from the 20 microns we measure, we decided to remove 15, hoping to be 5 microns from the surface after removing this. But actually then, when we measured distance again, we actually found that there were still 12 microns. And then we went back to the microtome, remove another 7 microns, and then at this point we were actually close enough with an estimation of 3 microns of the distance of the beginning of the cell of interest from the surface. And this is then when we decided, okay, this is now close enough for the following steps. So now essentially we went from a situation where all coordinates were not really known of our cell of interest, to a situation where now our Z problem is somehow solved, because we brought the sample now right under the surface of the block. So we have essentially solved the Z coordinate issue. But then we still need to solve the other two. So how do we define the XY coordinate? So here, the easiest way could be, if you don't have more sophisticated means, is simply to measure the distance of the cell of interest from edges of the blocks, for example. But because we were doing all of the confocal acquisition on a microscope that was equipped with two photon lasers, we decided to actually take advantage of this. And I don't know if you're aware of it, but there is some literature about near-infrared laser branding of tissues that was used, for example, for guiding serial block phase acquisition of brain slices and so on. So we decided actually to take advantage of the same system, but now to instead of burning and branding something within the tissue, we decided to just stay at the surface of the block and then perform branding there to be able to have some surface landmarks. And now you see the experiment is run here. So this is our cell of interest. You see now I have my confocal imaging focused on the block surface. So the cell of interest is slightly under the surface. So this is why it's not in focus. And now I have created some shapes that will define the branding marks that you will see now happening live. And you see this is actually marks that are engraved on the surface of the block. And now again, now for our system, we measure that to achieve this kind of marks without blasting the whole surface, we needed around 100 milliwatts, and also now the settings that we use are here. And then this allowed us to then go and brand some laser marks on the surface around several cells of interest within at the same height. So this is an example here. This was Carrel cell of interest in the smaller, and then we had another larger cell here. But because they're both more or less at the same height, then we branded the surface next to both. And now this then essentially also solves the whole problem, and now we have actually already defined a very well defined area that we can target by FIPSEM, and this is also very easy because this makes all the targeting of the FIPSEM very easy, and we can be very confident of not missing any part of the cell, because now we can go back after the laser branding and acquire another confocal stack of the region of interest, where we will be able to visualize the branding marks. And from this, then we can define exactly not only the surface deposition that we will do at the FIPSEM, but also the depth of the cell that is going to be that we need to cover at the FIPSEM. And this then will allow us to predict how
Hi, Paolo.
Can you hear me now?
Yeah, there you go. You were muted there for a minute.
Yeah. Okay, so then we can identify, I don't know actually where you stopped hearing me, but so we have from the light microscopy, we can recognize the shapes and then see how the cell of interest was oriented with respect to the shape, and then we can use the landmarks to actually define our volume of interest for the FIPSEMs, and then can be very small and targeted right on top of the cell of interest. Now, an important point. So I told you that the sample prep that we do for preserving the fluorescence is kind of a compromise. So we only use UA, and we also use low acrylation 20, which is a bit an exotic resin when you look at what is published about FIPSEM, and there's a reason for it. So low acryl is a bit softer compared to epoxy resins and Durcupand, which is one of the most used resins for FIPSEM, and this comes then with a price. So, and the price is that if you move around your region of interest on the block face during the FIPSEM experiment, this will result in pretty bad milling artifacts. So what we need is that we have to essentially define where the cell of interest will show up in our cross section of the surface, and to do this, in order to do this, we use the fluorescence map that we have, and this allows us to define, for example, now here the dashed line is the area of interest that we will acquire in our final FIPSEM acquisition, and as you will see, we have enough precision to actually, so that the cell of interest was contained in this image. And in this way, then, we will not move our window of acquisition for the entire volume, and this will result in a much, much better quality of the experiment. And then this is the result that we obtained for Carrel cell of interest. And I'm gonna play. This is the cell, where we see different organelles of the cell. So one interesting point is that we could also reconstruct the TICA of the cell that allows for a very good identification of the species, because typically taxonomists are using SEM and then the shapes of the plaques of the surface to identify the cell. But then we also have access to shape and measurement of the internal organelles. So for example, here we see the nucleus in blue, Golgi in yellow, and the flagella in green. Here we have the chromosome segmented in blue, the chloroplast in red, and some starch granules in white. And now here, this is a special organelles that is present in dinoflagellates. They are called trichocysts, and they're kind of small crystalline structures that can be extruded if the cells feel threatened by enemies, or if it just wants to be moving around. And what's interesting, for example, in this case, we were able to identify two classes of these trichocysts, of these organelles. Some were small and well aligned under the surface of the cell. And what we figured out that these were actually the ready to be extruded structures, whereas these longer blue structures were the ones that were probably undergoing formation, and they will be then later cut in smaller pieces that will be then sent to the plasma membrane. So now with this, segmentation is not only interesting to give us a nice 3D rendering, but it's also important because it allows us to measure the volumes of different organelles. And then what Karel has done was to compare the same cell under different conditions. For example, there were samples collected in the morning and in the evening, so right after the night, so when the cell was in the dark for some time, and then after the cell was with the light condition for the whole day. And then she's now looking at how the different volumes of the organelles are changing as effect of the light. Then another thing that we can do from this volume, as we have the light microscopy volume and we have the volume EM acquisition, and they are done exactly on the very same block. So nothing happens in between the two imaging modalities. We can actually now correlate very precisely the two volumes. So if you see, for example, here, we have a signal in magenta, which is the chlorophyll autofluorescence. And then you can actually see in the overlay that it matches very well with the chloroplast of the cell, as well as there is some autofluorescence in the nucleus that we cannot really explain at the moment, but these are all things that we are still investigating. And what can be interesting here is now, if instead of only looking at, I mean, this was kind of a low-hanging fruit because we already knew what to expect. We already knew that chlorophyll would be present in the chloroplast, right? So this was, I mean, it was a good confirmation in a way, but it was expected. But now when we can take different fluorescent channels and compare maybe also other fluorescent parameters, then we can be able to identify maybe specific autofluorescence of specific organelles within the cell, which is something we are exploring, and already preliminary experiments give very promising results. Now to conclude, I'm going to show you how this data can be visualized, and for this I'm going to show you a recording that has been done using MOBI. So this is a tool that was developed by some people at the EMBL to visualize and interact with large data sets. And here, this is work done by my colleague Martin Schorb, who has overlaid all of the different modalities in a single MOBI project that I'm going to show here. So now we start from the fluorescence in the full block, then we zoom in and we go at high resolution around the cell of interest. So this is now another high resolution stack where we see now the fluorescence appears nicer. We visualize a branding mark on the surface. And then we can further zoom in, and then we can overlay the EM volume. And now all the different channels and EM are all overlaid to have the final result. So another thing, so what I showed you so far was taking advantage of the autofluorescence of the plankton, but essentially we have shown in a couple of previous papers that the same trick can actually be used to look at fluorescent cells, so cells expressing fluorescently tagged proteins. In this case, for example, we had mammary gland organoids expressing H2B HM cherry, so to label the nuclei, and then you can see this is a fluorescence image of the block. So the fluorescence is preserved, and here are some details to show what kind of details the sample prep that we are using is able to give. So here you see, for example, a Golgi complex, some multivesicular bodies, mitochondria with the cristae, even some filaments, probably microtubules inside, and centrioles, so a centrosome here. And so not only then autofluorescence and fluorescent proteins, but also fluorescent dyes can be preserved. We have looked at Hooks, some FM dyes, and so on. So the only thing that we are not able to preserve is dyes that are binding to lipids, because during our free substitution protocol, the lipids are washed out. So yeah, here I also have some more characterization of the different fluorescent channels. So I'm not sure how relevant this is. So maybe we can save this for discussion if I'm a bit over time. If you're interested, so here I compared different fluorophores, how they would behave for the preservation in the block. And essentially, to make a long story short, every red fluorescence is better than GFP. And then with this, I'm at the end, and I would like to thank my colleagues in the electron microscopical facility. So Yannick Schwab is our head of the facility. Rachel is our operations manager. Mandy, Viola, Martin, and myself are people working on projects. Julia and Pedro are former members of the facility who have now left, but they were also very important in setting up these methods. Then the old Schwab team at the MBL, especially Karel, and the people in the light microscopy facility who have helped us setting up all the light microscopy imaging of the block, as well as the two-photon laser branding, and then people from Johan de Cell's lab who are involved in collecting the sample. And with this, I'm at the end, and I guess I can stop sharing. If you have any questions.
Thanks, Paolo. I think we can have 5 minutes for Q&A. I have one question, Paolo, about your beach experiments. So those dinoflagellate, they are seawater creature, right?
Correct.
Do you do anything special for high pressure freezing these aquatic animal in the? Do
No,
you
so-
add something
Yeah.
different to keep their osmolarity?
No, so we tried at the beginning to play around with different cryoprotectants and so on. So, but this didn't work very well, and especially not very reliably for all the cells we had in the sample. So what we figured out to be the best solution was to have a pretty high density of the cells and then simply freeze in seawater, so their natural environment.
Okay, all right. So the reason you use such high density block, you plan to reuse that block, right? You can go to other region, just keep going to select for different organisms.
Yeah.
Okay, all right, yeah.
And also to have enough mass to actually have the cell of interest within the block, because some cells were pretty rare. So for example, now the one that Karel looked at, I think we had only 3 in the big block where we have thousands of cells. So if the sample is not very abundant, or if the cell type is not very abundant, that you actually need to have high concentration.
Any other questions? Otherwise, I have another one for Paolo. about the preservation of the autofluorescence, are they preserved better than fluorescence proteins, in your opinion, or did they get any quench during from your embedding process, or not affected at all?
So I think the autofluorescence actually is better than fluorescence. And
Okay.
so the reason for this, I believe, is that especially like GFP, for example, is very sensitive to pH. And when we incubate with UA, which is very acidic, then I think this is quenching a lot the GFP fluorescence. But this is not the case for the autofluorescence, and therefore I think we can preserve this much better. So a very important point, as you asked this, especially for fluorescent proteins, is that even though there is, we are losing quite a lot of fluorescence during sample preparation, we can actually retrieve it quite well if we incubate in water. So we saw that if we incubate the block in water, we like get back up to 10 times brighter fluorescence on the block.
Wow.
But this, of course, doesn't go very deep. It's only probably the first 10, 20, 30 microns where there is still some effect of the water.
Good to know. Thanks. We do have a question in the chat. Janice is asking, how long was the
Also,
FIB
Alison
sen-
has her hand up.
You've already, yeah. Okay. If there's,
Hi, Paolo. Sorry. Yeah, beautiful. Thank you for that talk. Can you just explain a little bit more how you, the type of holders that you use when you're going from all of the different imaging modalities to keep your little bits of tissue, of cells and then resins semi-orientated, I guess?
Yeah, so what we use is the Leica IFS consumables. So you know if you are familiar with this, so it's kind of a revolver type of thing with 10 slots. So I'm simply using that. I place my carrier at the bottom of this, and in the end, I remove the carrier, so I have my 2mm, 2.5mm wide sample. And then the only thing I do is to shave off the very surface because it has some concentric rings which would diffract the light, and that's not good for this. And then the other thing I do is to manually trim the block surface to get an asymmetric shape, but very roughly with a razor blade.
And when you're trimming it, you're holding it in the microtome just with the traditional block. You're not sticking it to a pin.